we all grow up with a neat version of human evolution. Neanderthals in Europe, Denisovans in Asia, modern humans in Africa. Then, somewhere along the way, a quiet assumption hardened into something much bigger, that African populations were the least mixed, the least interrupted, the closest thing to an untouched source. It sounded scientific, it sounded careful, but in hindsight, it may have been one of the biggest blind spots in human origins research, because that idea was never based on overwhelming evidence that archaic mixing did not happen in Africa. It was based, in large part, on absence. No clear archaic reference genome from tropical West Africa. Very few well-preserved fossils, almost no ancient DNA from the places where the deepest parts of our story may have unfolded. And absence has a way of turning into confidence when a field does not yet have the tools to see what it is missing. Here is the part that matters. Tropical heat is not just uncomfortable for archaeologists, it is destructive. Bone breaks down faster, DNA fragments faster. Humidity, microbes, and acidic soils do not simply hide evidence, they actively erase it. So for years, Africa, especially tropical Africa, looked quieter than Europe and Siberia, not necessarily because less happened there, but because much less survived in a readable form. That is a very different claim, and once you see that distinction, the old certainty starts to wobble. That is the first pattern interrupt in this story. The missing fossils were not proof of purity. They may have been proof of terrible preservation. And then there was Iwo Elaru. In Nigeria, at a site called Iwo Elaru, researchers studied a skull from a burial dated to the terminal Pleistocene, roughly the closing stretch of the last ice age. By date alone, you would expect something recognizably modern. Not necessarily identical to people today, but clearly within the recent human range. Instead, the skull kept bothering researchers because its shape did not sit comfortably where it was supposed to sit. It showed what scientists sometimes call a mosaic pattern. In plain language, that means it looked like a mix of traits that did not neatly belong together. Some features looked more recent. Others looked unexpectedly archaic, as if a much older form had somehow lingered far later than expected. One analysis concluded that Iwo Elaru fell outside the normal range of variation for other terminal Pleistocene African samples and argued that its anatomy was consistent either with admixture from archaic humans or with the survival of populations retaining more archaic features until very late. That is not a minor wrinkle. That is a timeline problem. And this is where the story gets interesting, because Iwo Elaru did not give researchers a clean answer. It gave them a warning. Not proof of a lost species, not a solved mystery, a warning, a signal that Africa's population history might be much more layered than the simplified model allowed, that in some regions deeply divergent human groups may have persisted alongside populations we would call modern, that physical differences seen in the fossil record might reflect isolation, survival, mixture, or some combination of all three. Even the researchers were careful here. The point was not certainty. The point was that the old assumption no longer felt safe. So now the problem sharpened. If fossils were scarce and ancient DNA was mostly gone, how do you test whether archaic humans contributed ancestry to living Africans? Normally, you would compare modern genomes to a known archaic genome. That is how Neanderthal ancestry became visible. That is how Denisovan ancestry became visible. You need a reference, a target, a known ghost. But what if the ghost left no usable bones? What if the only thing it left behind was scattered code inside living people? That was the challenge, and solving it required a shift from classical comparison to something much stranger, teaching an algorithm to recognize interbreeding without ever being shown the vanished population itself. So if the fossil record could not give them a face, researchers had to look for behavior instead, not behavior in the usual sense, genetic behavior, because archaic interbreeding leaves behind a very specific kind of signal in DNA. It does not just create random weirdness. It creates stretches of sequence that look unusually old, unusually distinct, and oddly clustered compared with the rest of the genome. over thousands of generations. Recombination chops those stretches into smaller pieces, but the statistical fingerprints can still survive. The challenge is separating those fingerprints from everything else that also makes DNA look strange: mutation, drift, population splits, bottlenecks, and plain old noise. And that is where the algorithm enters the story. Instead of asking, do these genomes match Neanderthals?, researchers asked a more flexible question. Do any regions in these genomes behave like introgressed DNA should behave, even if we have no fossil genome to compare them to? That is a huge shift. Normally, ancient admixture studies lean on a reference. You have a Neanderthal genome. You compare modern people to it. Shared segments stand out, but in West Africa, the suspected source population was unknown. No confirmed genome, no clean fossil target, so the method had to work blind. What the model looked for were combinations of clues, segments that were unusually divergent from typical modern human variation. Segments carrying clusters of derived mutations that were rare or absent in an outgroup population. Segments whose internal structure suggested they were inherited as ancient blocks rather than assembled gradually through ordinary variation. In other words, not one magic marker, but a pattern, a signature. That is why machine learning mattered here. A human can look at one statistic at a time. The algorithm can be trained on many simulated demographic scenarios and learn which combinations of statistics most often correspond to archaic introgression. It is less like asking a computer to identify a single fossil bone and more like teaching it to recognize the shape of a crime scene after the body is gone. And when that kind of method scans living genomes, it can find candidates that would be nearly impossible to spot by eye. Now here is the pattern interrupt, because this is the visual that makes the whole idea click. Imagine a graph of allele frequencies. On one axis, you track how common a variant is. On the other, how many variants fall into each frequency bin. Under some demographic histories, you expect one shape. Under introgression, especially when an older population contributes DNA that later drifts through a descendant population, you can get something more revealing, a U-shaped spectrum. That means you see an excess at the low frequency end and also an excess at the high frequency end, instead of a smooth middle-heavy distribution. In plain English, some archaic derived variants stay rare, scattered like broken fragments. But others rise to high frequency because they arrived together on inherited haplotypes, drifted upward, or were favored by selection. That split pattern is a clue that you are not just looking at ordinary variation inside one clean lineage. You may be looking at genetic material that entered from outside it. So the graph matters because it answers a deeper question. Are these variants behaving like part of the same old human population, or like pieces imported from a long-separated branch? The algorithm's job was to weigh that question across genomes from West African populations and ask over and over where the data fit better, with a model of simple population history or with a model that included ghost admixture. And this is where the story turns from suspicion to shock, because once the model started flagging these regions consistently, the signal was not tiny. It was not some fringe anomaly hanging on one weird chromosome or one isolated family line. The pattern pointed to a real ancestral contribution from a deeply diverged population, one that had never been directly sampled, never sequenced, and possibly never identified in the fossil record at all. At that moment, the ghost was no longer just a metaphor. It became a measurable population history event hidden in modern code. And the size of that signal was large enough to force a much bigger question. If this lineage was invisible in the bones, how much of ancient humanity has been hiding in us the whole time? Now comes the part that made this story travel far beyond genetic circles. When researchers applied this reference-free approach to living West African genomes, the signal pointed to something substantial. These populations appeared to carry ancestry from an unknown archaic population that split from the lineage, leading to modern humans long before recent human populations diversified. In the paper, the estimated contribution was not framed as one neat universal number. The model suggested roughly 2% to 19% ancestry from this ghost population across the sampled West African groups, with popular retellings often rounding that into a memorable middle figure like 7%. That middle number works as a headline, but the real result is more nuanced and more interesting. That distinction matters, because when people hear 7% ghost human, they often imagine that scientists found one person who was 7% some lost species, almost like a modern hybrid walking around with a visible label. That is not what this means. This is a population genetic estimate. It is about how much ancestry in the model populations traces back to an unsampled archaic lineage, averaged through deep time, recombination, and demographic history. It is a statement about inheritance patterns and populations, not a dramatic claim that a living group is secretly part other species in some simplistic sense. And the ghost itself was old. The inferred source population appears to have diverged before the split between modern humans and Neanderthals in the model used by the study. In plain language, this was not just a slightly isolated neighboring group. It was a deeply separated human lineage, ancient enough that, if we had its bones and DNA in hand, it could fundamentally reshape how we picture Middle Pleistocene Africa. Then there is the second twist. This ghost population was not necessarily tiny or marginal. The authors argued that the archaic source may have remained around long enough and mixed strongly enough to leave a broad signal in present-day genomes. That implies something important. We are not looking at a brief accident, some vanishing encounter with a handful of isolated individuals. We may be looking at a thriving population that coexisted with ancestors of living West Africans and contributed meaningfully to their gene pool. A lineage invisible in fossils but not small in consequence. And once you accept that possibility, the mental picture of ancient Africa changes immediately. Instead of one clean ancestral population gradually becoming modern, you get a continent inhabited by multiple human groups, some more closely related, some more divergent, sometimes isolated, sometimes reconnecting. Not a straight line. More like braided channels crossing, separating, and crossing again. The genome starts to look less like a family tree with tidy branches and more like a record of repeated contact among lineages that did not all leave fossils we can identify. There is also a reason this result hit so hard culturally. For decades, the public story of archaic admixture was geographically lopsided. Neanderthals belonged to Eurasia. Denisovans belonged to Asia and Oceania. Africa was often treated as the clean origin point, the place before the mixing began. But this result suggested something almost opposite, that Africa may have preserved some of the most complex and least visible admixture events of all, precisely because the fossil and ancient DNA record is hardest to recover there. The old purity picture did not just weaken, it started to collapse. Now, to be careful, this is still an inference from modern genomes. Researchers are not claiming they have excavated a skull, named a species, and closed the case. Competing explanations and alternative demographic models have been debated, including models with deep population structure inside Africa rather than a single cleanly separate archaic species. So the strongest honest version is this. The genomes contain a robust signal of ancestry from a deeply divergent lineage, and exactly how that lineage should be classified remains part of the scientific conversation. But even with that caution in place, the reveal remains extraordinary. A vanished population, no reference genome, no confirmed fossil match. Still detectable. Not because we found its bones, but because its descendants are carrying fragments of its history right now. And just when that sounds like the deepest layer of the mystery, another clue appears from a completely different part of the genome, one that does not track broad ancestry across all chromosomes, but a single paternal line that seems almost impossibly old. That clue is called A0. If the ghost admixture result suggested that something ancient had survived inside the autosomal genome, haplogroup A0 added a very different kind of clue. Because this was not a broad ancestry signal spread across all your chromosomes, this was a Y chromosome, a direct paternal line. One branch passed from father to son, over and over, like a single thread surviving through immense time. And when researchers identified Aasiarowan in living men from Cameroon, that thread seemed shockingly old. To understand why that mattered, we need one quick reset. A Y chromosome haplogroup is not a species. It is not proof that someone belongs to an archaic population. It simply marks a branch in a paternal lineage tree. But sometimes a branch is so deep, so unexpectedly distant from the others, that it tells you the tree itself may be older and more complicated than people realized. That was the reaction to A7. Compared with previously known Y lineages, A0 sat further back, on a branch that appeared to split extraordinarily early. The estimated age of that divergence pushed far deeper than many researchers expected from living human Y chromosomes. And because it was found in Cameroon, it immediately fed into a broader realization that Central and Western Africa may preserve some of the oldest surviving genetic diversity on Earth. Now, this is where weaker storytelling usually makes a mistake. It turns A01 into a dramatic headline like, "Scientists found an alien Y chromosome," or, "This proves a lost species survived in Africa." That is not the honest version. AU does not prove the exact same ghost population detected in archaic admixture models. What it does is more subtle and in some ways more powerful. It shows that extremely ancient human lineages can persist in living populations far longer than a simplified model would suggest. It warns us that the surviving genetic landscape in Africa contains branches deep enough to challenge tidy narratives. And it reinforces the idea that if we assumed the African record was genetically straightforward, we were probably underestimating how much diversity had once existed there. In other words, AO is not the answer to the ghost. It is corroborating atmosphere. It is another sign that the old map was missing pieces. And think about how unusual that is. By this point, we already have one line of evidence saying modern genomes may contain ancestry from a deeply divergent, unsampled population. Now, from another direction entirely, we also have a paternal lineage that seems to reach back into unexpectedly ancient structure. Different signal, different method, different scale. But emotionally, they point the same way, backward, into a version of human history that looks less settled the more carefully you examine it. There is also a deeper lesson here about how genetics works in storytelling. People often want one discovery to do everything. One skull that proves the case. One genome that closes the argument. One branch that names the mystery. Real science almost never works that cleanly. More often you get overlapping hints, an anatomical anomaly here, a statistical signal there, a lineage tree that suddenly grows an older root than expected. None of them alone gives you the whole vanished population. Together, they start changing what kinds of explanations are plausible. That is exactly what A-tensor does in the story. It widens the zone of possibility. It tells us that ancient population structure in Africa was not shallow, not simple, not exhausted by the lineages we first learned to recognize, and once that door opens, the ghost admixture signal stops feeling like an impossible outlier. It starts feeling like part of a larger pattern, which brings us to the final step. If these hidden lineages can be detected without fossils, and if the genome keeps preserving clues long after the bones are gone, then we need a way to track this kind of evidence clearly, not just marvel at it. Not just repeat the headline. We need a framework. So in the last part, we build one. So how do you think clearly about a story like this without getting lost in hype? Because once you hear a phrase like unknown human species hidden in our DNA, two bad reactions usually take over. One is blind excitement. The other is instant dismissal. But the real story is better than either of those. It is not magic and it is not nothing. It is a case study in how modern genetics can recover missing chapters of prehistory from patterns that survive in living people. That is where the archaic admixture search framework comes in. Here is step 1. Look for the assumption being challenged. In this case, the assumption was that African populations were free of archaic interbreeding, or at least mostly untouched by it. That assumption sounded stable because the fossil and ancient DNA record was thin. But that is exactly the point. A thin record does not only limit what you know, it can also make a false picture look clean. Step 2. Ask what kind of evidence is missing and why. That matters more than people think. In tropical Africa, heat, moisture, microbes, and time destroy the very materials researchers would normally use to test these questions. So the absence of archaic fossils is not neutral. It has a cause. Once you understand that, you stop treating missing evidence as evidence of missing populations. Step 3. Identify the anomaly. Every major shift starts with something that does not fit. Here, one anomaly was anatomical. Iwoeleru, a late skull with features that seemed unexpectedly archaic. Not a solved puzzle. A pressure point, a sign that the standard timeline might be too simple. Step 4. Follow the method, not the headline. This is where most people skip ahead too fast. The key breakthrough was not that scientists suddenly found a bone and named a species. The breakthrough was methodological. They used machine learning and population genetic modeling to detect introgression without a reference genome. That is the real innovation. Once you understand the method, the claim becomes much easier to evaluate. Step 5. Look for statistical fingerprints, not dramatic labels. What matters are things like unusual divergence, inherited blocks of sequence, allele frequency patterns, and models that fit admixture better than simpler demographic histories. The genome is not shouting a species name, it is leaving behind a pattern of probabilities. Your job is to ask whether those probabilities consistently point in the same direction. Step 6. Separate signal strength from interpretation strength. This is crucial. The signal may be strong while the interpretation remains partly debated. In this story, the evidence supports ancestry from a deeply divergent lineage in West African populations. But whether that lineage should be called a distinct archaic species, a structured ancient population, or something in between, is still a matter of interpretation. Good science lives in that distinction. Step 7. Look for converging clues. One clue can mislead. Several independent clues are harder to ignore. Here you have the preservation problem, the fossil anomaly, the algorithmic detection of ghost admixture, and the extremely deep Y chromosome branch A00. None of these alone gives you a perfect portrait. Together, they tell you that ancient Africa almost certainly held more human diversity than the old model allowed. And that leads to the final takeaway. The biggest discovery here is not just that a ghost lineage may have contributed ancestry to living people. It is that the human story is still recoverable even when the bones are gone. That is an extraordinary shift. For most of scientific history, if a population left no fossils and no recoverable ancient DNA, it was effectively lost. Now, that is no longer completely true. Fragments can persist in modern genomes, algorithms can detect patterns too subtle for the eye, and lineages once erased from the ground can remain partially visible in code. So the next time you hear that human evolution is settled, remember this story. A continent once assumed to be genetically pure may preserve some of the deepest admixture of all. A vanished population may still be measurable without a single confirmed fossil genome. And the oldest chapters of our history may not be buried in stone. They may be walking around inside us. Hit like if AI discovering new human species amazes you.